Okay, go for it.
Hi, everyone. Hi, everyone. Thank you for attending this IE2 seminar. Dr. Maggie Sogin is our speaker today. She got her PhD through the Department of Zoology and the Hawaiian Institute of Marine Biology at the University of Hawaii in Manoa. She then worked as a postdoctoral scientist and later a project director at the Department of Symbiosis at the Max Planck Institute of Microbiology in Bremen, Germany. Currently, Dr. Sogin is an assistant professor at the Department of Molecular and Cellular Biology at the University of California, Merced. Her lab studies host microbial interactions and symbiosis. Some of the host species in her lab works with are seagrasses, sea anemones, and insect pests of the almond trees. Thanks again for attending, and whenever you're ready, Maggie.
All right. Thank you. Thank you, Caitlin, for the introduction. And I hope everyone in the room can hear me okay. And everybody else in Zoom land can also hear me. I want to thanks again for the invitation. And I have to say, like, I don't always enjoy the remote seminars, but in this case, I'm happy a remote seminar as I am currently battling COVID. So bear with me in this sort of presentation. The title of my talk today is about metabolites and how they shape host-microbe interactions in the sea. And I want to give you an overview about how I think about microbial interactions and symbiotic associations and present with you two stories from my lab that I've done as a postdoc and now that I'm working on with a group of researchers here at UC Merced. So I first like to frame my talk by thinking about this idea that we live in this symbiotic world. And of course, as you might guess from the title of my talk, I focus mostly on symbiotic interactions of microbial symbionts, but of course, symbiosis could be between any organisms. And so if we think about symbiosis all around us, we can look at this elephant and the bird, and we know that there's some sort of mutual benefit where the bird is picking off the insects that are on the elephant's back and giving it a nice bath. This was for a long time what people thought about when they thought about symbioses, about having these interactions. We have symbioses in our daily lives when we think about how we cultivate or agriculture species, like where I am in the Central Valley, we grow lots of almonds, the almond trees persist, and we get the benefit of being able to eat the almonds. But of course, microbes are what I'm here to talk to you about today, because they really define almost all different types of life on Earth. Everything from these organisms that I've had the pleasure to work with as a researcher, both as a PhD student, but also as a postdoc, and now as an assistant professor. And one of the questions that I'd like to sort of explain to you was about what I consider to be a symbiont. And so what does this really mean? So of course, symbioses can range from simple associations to complex communities. And simple associations, like within this gutless worm that lives in marine sediment, we know that the worms, they only host one or several different types of symbionts. And so they're really simple in terms of like the interactions that they have. Whereas in humans or in coral reefs, for example, we know that these animals actually have these diverse microbial consortia that can have interaction benefits that range from different types of associations. So for example, these microbes can range from everything from being mutualistic, where there is a net benefit to both partners, or they can become pathogens. And these interactions can be between different types of microbes. So you have microbe-microbe interactions, but they can also be between eukaryotes and microbes. And importantly, these cellular interactions are often mediated by the exchange of small compounds. So corals might provide some resources to the microbial community, and the microbial community might then provide some resources to the coral host. And these interactions can change depending on the environmental conditions that the organism persists in. So for example, some environments might lead to the relationship being mutually beneficial, while other environments would lead it to being more pathogenic and disruptive for the actual symbiosis. So one of the questions that I am particularly interested in studying in my lab is how do environmental and biological processes shape microbial associations? I hope So I say on the chat that you lost the video, but you can still hear me okay. Yes?
Yes.
Okay, just want to make sure. As opposed to going through the entire talk, that video will be heard. Okay.
Can you hear us?
Yeah, I can actually. It's not
Okay.
too distracting, but I think it's fine.
Don't worry, you're fine. We can see you and hear you great.
All right, awesome. Okay. So this is the fundamental question that I think about in my research in my group, is how these interactions between hosts and microbes in the environment are shaping microbial associations. And so we can think about these associations with this sort of triangle. And this is actually the same triangle between host, microbe, and environment that we think about when we look at disease triads in our microbiology class. We have these associations where the host is interacting with the microbes are interacting with the hosts or the environment are impacting both of these associations. And we know that these associations, these symbioses, also can impact the environment. And the interaction between these different organisms sort of dictates if the association is going to be beneficial to pathogenic, and what it might mean for everything from local interactions on coastal communities to ecosystem scale processes. So what we do in our lab is to actually try to not get up into the weeds about these different interactions, but to try to step back and take a more holistic approach. I kind of like this cartoon because it makes me think that, like, if you are only studying these interactions from one lens, from the host lens or the microbe lens or the environment lens, you're probably going to miss some major part that this is in fact an elephant. And so we combine different types of approaches, including metagenomics and metatranscriptomics and metabolomics, and we integrate these with systems level approaches to understand complex host and microbial metabolisms. So this kind of brings me to the outline of my talk today. And I really want to tell you about two different stories. The first story is work that I did as a postdoctoral researcher at the Max Planck Institute in Nicole Dublia's group. And the second is about the work that we're doing here to try to introduce students at UC Merced to marine ecology research. And I could tell you a little bit about the challenges of trying to do marine ecology in the Central Valley of California. But first, I'd like to start with this title that we have titled our story is really the sweet spots of the sea. And specifically, the title of this particular section of the talk is that sugars dominate the marine rhizosphere. So I like to make the joke that this is the punchline of my story. In case you miss all the details, this is the main message that I want to tell you. And as a postdoc, I got to work in this really beautiful ecosystem in Mediterranean Sea, which is founded by this seagrass species called Posidonia oceanica. And what's really incredible to me about this particular type of seagrass is that it is considered one of these blue carbon ecosystems because it has this large capacity to sequester CO2. So we know seagrasses that they cover less than 10% of the ocean surface, but they cover less than 1.1% of the ocean surface, but they sequester up to 10% of marine organic carbon. And this is really remarkable because it can sequester carbon faster than tropical rainforests. So one of the questions that we have in our group and that others are asking is what makes these seagrasses such efficient carbon sinks? And I have to admit to you that the reason why I got so excited about studying seagrasses and thinking about these interactions really stems from the evolutionary history of plants, because I am self-admittedly not a botanist. And I just found that this is so fascinating to think about how symbiotic associations might be evolutionarily conserved. So we know that land plants, they evolved from sea algae, and they went up and they colonized the Earth's surface. And then there was a lineage of angiosperms that fall into that monoclot lineage that was able to repopulate the sea on multiple occasions. So there's about 3 separate independent events where seagrasses transitioned back from going on land and into the ocean. We know on land that these plants, they release secondary metabolites and other types of compounds to their soils to attract a beneficial community of microbes. And one of the things that I was interested in is, can we actually see similar patterns in the marine world? And can we begin to characterize these types of interactions in the sea? So this brings me to tell you a little bit about the star of our show, which is Posidonia oceanica. As I said, I got to go to the Mediterranean quite a few times and work off of the coast of Italy. It's not shown here on this map, but there's this beautiful island called Elba. We went and there many times. We got to sample different types of seagrass beds, well, the same type of seagrass bed, but in different locations. And in parallel, we also sampled the different types of gelato that was being served in Elba. And it was a really great place to go and to be able to do field research in this really, really beautiful country. And the work that I am going to tell you a bit about in our findings was recently published this past year in Nature Ecology and Evolution. So we were going to Elba and we were collecting pore water samples for metabolomic analysis. And so you can think of the pore water as really being that water that's in the space between sand grains within a sediments. And we were collecting across different sediment depths, trying to understand what is the metabolite composition of this particular ecosystem. And we were bringing these samples back and we were trying to inject them onto our GCMS in order to characterize the presence and abundance of these small molecules. But every time we injected these samples onto this instrument, we actually really didn't see very much signal at all. And we couldn't really characterize what the metabolite composition is. So if you are a mass spectrometrist, and I don't know if any of you are in the room, but you probably already know part of the reason is that salt and water are particularly bad for mass spectrometers. And so during the course of my postdoc, I developed a method that really is just a few quick derivatization tricks that would allow us to analyze the metabolite composition of salty samples in a much better fashion than had been previously done before with our GCMS method. And I can just tell you the benefits of GCMS is that we can analyze a lot of samples in one day. We only needed about 1 milliliter of sample volume, which allows us to do more high resolution sampling in terms of spatial or temporal scales. And really nicely, GCMSs are considered the workhorse for metabolomics facilities, and they're present globally. And so you can really apply our method no matter where you are if you want to try to understand the metabolomics of a marine habitat. So we came back to the lab, we took our samples. With our new method, all of a sudden we're able to detect peaks in our pore water samples. And actually, this method has been really good for us in terms of like looking at culture experiments of microbial communities with salty media, and of course, metabolomics for salty habitats. So if we go back into our ecosystem, we continue to collect samples for metabolomic analysis. We were looking at samples both inside at the edge and outside of the seagrass meadow, with the goal of trying to say what is different in terms of metabolite composition underneath a vegetated sediment versus in a non-vegetated sediment. And when we take a look at that data, the most abundant metabolite across our different pore water profiles was this compound sucrose, which is a simple sugar. And actually, it was quite concentrated where we were able to find. We're able to detect sucrose up to millimolar concentration levels within this habitat, which is really quite remarkable when you consider the fact that sucrose, as being this energy and carbon resource that is really easily consumed by microbial communities, it really shouldn't be there, because these simple sugars are regularly consumed by microbes. We learn this, this is a fundamental aspect that we teach in our introductory microbiology class. So I want to tell you about the work that we did to both show that the seagrasses are excreting this sucrose molecule into the rhizosphere. And then I'm going to tell you a bit about why our data show that the microbes are simply just not being able to consume them. So our first approach towards trying to understand what the temporal dynamics of the sucrose cycling within its habitat was to actually do a 24-hour sampling effort. And so this is the meadow that we found that allowed us to actually snorkel and install these pore water lances into the sediment. So this is me sort of swimming along, and I have these lances that we installed at fixed positions in the seagrass meadow, and we sampled across a 24-hour time period. And I just want to highlight that this work was really made possible because DOMA helped keep me awake and keep me going during the 24-hour sampling push. In addition to collecting pore water, we also collected individual seagrass plants, and we preserved them for metabolomic analysis so we could characterize how the plants might be using sucrose in that same temporal time period. And when we take a look at the data, the first thing that we see is that sucrose concentrations change throughout the day in both the seagrass leaves and the pore water samples. And so I'm going to show you sucrose abundance on the y-axis and the time on the x-axis. And we see this being significant both in the leaves and the pore water space. And I can tell you that this was not significant in either of the seagrass rhizomes or the root tissues. And so what this tells me is that during the day, the plant is photosynthesizing and producing sucrose. And at the night, the photosynthetic processes are dropping off. And we see that sucrose is only being really released significantly during the day to keep up that levels and accumulation. The second thing that we measured was to look and compare the pore water profiles in comparison to the typical depth of the Posidonia root system. And when we take a look at that data, we see that the highest concentrations of sucrose are within that same region of the roots themselves. So between that -5 and that -20 centimeters underneath the sediment surface. And finally, I got to work with and learn from Benedict Gear, who was a PhD student in the lab at the time, to conduct mass spectrometry imaging measurements to look at the molecular distribution of sucrose across a root section. So what we can do with this technique is we can section a typical seagrass root, and we see that it has similar plant root structures as a terrestrial plant. So we have the stele, which is in the center here, the cortex, and then the rhizodermis of the seagrass root. And then we can spray it with a matrix, and we can collect a 3D map of any ion that our mass detector can actually detect. And in this case, I'm showing you the map or the distribution for the ion that corresponds to sucrose in this case. And what we observe in this analysis is that sucrose generally is relatively high in comparison to other regions of the plant inside of the cortex, and that it really increases on the plant surface. And so what this suggested to us is that the sucrose can be stored in the root over longer periods of time and then pushed out to the rhizodermis, where it then can leak into the environment itself. So the next thing I want to tell you a bit is about the microbial consumption of this really important resource. And we chose to look at this through two different approaches. One, explore the sequence-based community dynamics of the microbial community that's within sediment cores inside the seagrass meadow, and the second through incubation experiments. So we went back out into the field and we collected these root cores, and we sectioned them at the region where we see the highest concentration of sucrose within the environment. And we extracted these sediments for both DNA and RNA in preparation for both metagenomics and metatranscriptomic analyses. So we did this typical pipeline where you take your DNA, you sequence it, you reconstruct your microbial genomes, and we annotated the gene content using the Caszyme database to really explore the ability of the microbial community to degrade sugars within the sediments. And then we mapped our transcriptomic reads back onto those microbial genomes to explore expression within the habitat. So I used to, in these talks, sort of show expression data for these different microbial communities, but I actually think it's much more remarkable if I just sort of tell you the punchline, because I could show you metabolic maps for these microbes. And I could show you how the relative proportion or investment of the communities are in terms of degrading sucrose to other sugars. But really the take-home message that from our transcriptomics analysis is that most of the microbial taxa that are located within the sediment layer are not expressing the genes to degrade sucrose. So just to let that sink in, despite in this environment having this large amount of sucrose as available for their consumption, most of the community is not expressing genes for the import or breakdown of this important resource. However, I use the word most because there are a few exceptions that I will come back to at the end of my talk where we see this is not the case. So the second thing we wanted to do is to use sediment incubation experiments to see if the communities have the ability to degrade sucrose and to measure metabolic rates within the sediments. So what we did is, because we know that the upper sediment layers are oxic during and they have oxygen in the environment, and we know that oxygen is rapidly depleted, we divided our sediments into the oxic portion of the sediment core and the anoxic portion, and we split them up into our incubation bottles. We also looked at both sediments that were collected from inside of the meadow and sediments that were collected from the edge of the meadow. And we dosed this incubation bottles with 50 micromolar of 13C sucrose to reflect that environmental concentration of sucrose that we see in the environment. And we measured sucrose consumption over time. And I want to show you the plots of that data where I'll have time on the x-axis of these sediment incubation experiments. And then on the y-axis, I have 13C sucrose concentrations. And once again, it's split between inside the meadow and at the edge of the meadow and between oxic and anoxic conditions. And when we first look at this data, this is what we sort of found, where the highest rates of sucrose consumption actually were in this anoxic sediment layer. And sucrose was removed within the first 12 hours of the incubation experiment. But this was very confusing to us because this was where we observed the highest sucrose concentrations in the field. And if this incubation rates were to be true, it would have to mean that the production of sucrose far outweighed the consumption of the sucrose for us to actually measure that accumulation within the environment. So we kind of went back and we scratched our heads. And one of the questions that we sort of posed to ourselves and our group of researchers that were in our department at the time is, could there be presence of other factors that actually inhibit or limit the respiration of sucrose within this environment? And this is where it's really fun to work with large groups of people and lots of PhD students, because Patrick came to us and he said, maybe there are other compounds like phenolics that could actually restrict the microbial metabolism in this environment. We know that seagrasses produce phenolics, including within our Posidonia oceanica tissues. And we also know that if we look at our dissolved organic matter profiles from this environment, we have higher concentrations of phenolics like caffeic acid inside the meadow than at the edge or outside of the seagrass meadow. So we repeated these incubation experiments, and this time we added phenolics to incubations. And what we see is very quickly that while in some conditions, like under oxic and at the edge of the seagrass meadow, the phenolics had no effect on the respiration of sucrose by the microbial communities. Under anoxia, all of a sudden, we see that their phenolics are limiting microbial respiration. So if we contextualize our results and we think about how under normal conditions, the sucrose, the microbial community that is within that sediments can actually degrade the sucrose, and the sucrose pool would rapidly shrink in the environment. But under meadow conditions where there is anoxia and the presence of seagrass produced phenolics, all of a sudden the microbes cannot use the sucrose as easily in their metabolism, and we see that sucrose pool begin to grow. So what we propose is that this is a mechanism that could prevent the metabolism of sucrose under certain conditions, and therefore represents a highly selective environment for this community. So why would seagrasses deposit their sugars into their soils? And this was a question that I, for a long time, was regularly asked when I presented this work. And we have several different ideas of why this could be happening. So first, we predict that this could represent some sort of overflow valve. On land, terrestrial plants will release excess photosynthate through their stomata, and they'll open them up into the environment. But seagrasses, because they have these evolutionary adaptations that allow them to live in the sea, they actually lost their stomata when they transitioned back into that ecosystem. So therefore, they have to put their photosynthate somewhere into their soils. So one of the things that we thought about was the presence of other electron acceptors like oxygen or nitrates. But of course, in the sediment ecosystem, I've already told you that oxygen concentrations are quite low in this habitat. And the nitrate concentrations are also relatively low. And so that would prevent the community to actually use the sucrose in their metabolisms. However, sulfate concentrations are high. And the issue with having a high sulfate concentrations in a microbial community that can use it to respire sucrose is that you would allow for fermented pathways to occur, and the sulfite would build up within these sediments, which is usually toxic to seagrass meadows. But of course, if you block that metabolism with phenolics, you wouldn't see that toxic effect of the sulfites in the sediments. The next thing is that we believe that these processes are likely conserved across aquatic rhizospheres. We didn't just go to the Posidonia meadows, but we also went to different types of seagrass meadows off of Belize, and we see similarly high concentrations of sucrose underneath two other different types of seagrasses, and we see high concentrations within a mangrove forest. We also find that this community recruits a specific community of microbes within the seagrass meadows themselves. So the composition of the microbial taxa that are underneath the seagrass meadow are quite different from at the edge or outside the meadow. And in fact, they have a relatively lower diversity. And the community of microbes includes the presence of some really interesting taxa that can degrade sucrose in the presence of phenolics, like a few key heterotrophs, some anaerobic sulfate reducers, and a couple of nitrogen fixers. Sorry, I'm not sure if people in the room need something from me. Is that okay?
Hello, what's up?
Can you still hear me?
Yeah, we can hear you.
Okay, sorry, I heard some background, I wasn't sure.
Closing the door.
Okay, sounds good. So I think what we come to is that globally, if we extend our calculations and we estimate how much sucrose is buried underneath seagrass meadows, it actually is quite high. And this is particularly, really represents this complex factors in terms of abiotic and biotic conditions that would lead to the accumulation of sucrose in this pool. And of course, if you begin to disrupt the seagrass meadow, it could represent a massive release of CO2 into like a one-time pulse into the atmosphere. So some things that we have in the back of our minds in terms of moving forward. And I just want to give you some ideas for next steps that we're going to continue with the seagrass system before I transition and tell you about the other story. I currently have a postdoc who I'm co-mentoring with folks at UC Davis, and she's really interested in what the role of these compounds are in carbon burial. Furthermore, my PhD student, Diane, she's really interested in how sediment microbes might promote seagrass growth and what other metabolites are shaping that microbial community. We know that, of course, seagrasses are producing other things than just sucrose, and we can see that there's a number of metabolites, including compounds that are involved in microbial metabolisms within the seagrass roots themselves, I think are interesting targets for investigation. And finally, one of the questions I'm particularly interested in is what is the fate of these interactions in the face of global climate change? So I started my lab in January of 2021, and I came to the Central Valley with the idea of using marine ecology tools to explore microbial interactions. And I first want to give you a little bit of an introduction to UC Merced because it's a relatively new university. The campus was established in 2005. It's considered the first university of the 21st century. We are located within the Central Valley, which is really in the heart of California. So we're about 2 hours east of San Francisco and about 2 hours away from Yosemite Valley. We have approximately 8,500 students, and the majority of them are Hispanic origin, and the vast majority, over almost 2/3, are, 3/4 are first-generation students. So this means that the university, and particularly my lab, has a special call, and specifically we want to recruit diverse students to my research group and give them confidence to pursue careers in STEM. And particularly, I've been thinking a lot about how to introduce first-generation students to research here on campus. So in order to engage undergrads at UC Merced in marine ecology, we would need to go out to Bodega Bay in order to do our field collections. This is where primarily our seagrass work is being conducted so far. And it is a really nice place to go and visit, and I love working out there, and it's been really fun. But it's also a 6-hour round trip, which makes it relatively inaccessible for many of the students who are located here on our campus. That's because many of our students, they have jobs that need to have as a part-time basis, or that they go home on the weekends to visit with their families. And so I've been thinking about different ways of engaging students at research, and we decided to begin to work with Aptasia, which is a sea anemone that has been typically used for understanding coral algal symbiosis within the community. And I want to tell you a little bit about work that 3 undergrads, Sam, Aaron, and Crystal, have been doing in the lab in conjunction with my PhD, or my technician, Sophie. so far in the last 1.5 years since we arrived to UC Merced. So first, I want to tell you a little bit about the power of Aptasia. So there is a global community of researchers that have begun to establish Aptasia as this model system for exploring coral algal symbiosis. They are incredibly easy to cultivate, and maybe too easily. People think of them as these aquarium pests that once you have an infection in your aquarium, it's really hard to get away. And this is really convenient for us because we can actually keep them in just Tupperwares in the lab. We only have to feed them twice a week, and we have to change their water, and it's super simple to propagate the population. You just cut them in half and then grow 2 new individuals. There is this rich repository of resources that's available because this is being an emerging community of researchers who are beginning to work on them. And lastly, what I want to tell you about is how we can manipulate their microbiome to explore host microbe interactions. So the primary question that I have with this project is, how does metabolic cross-feeding within a microbiome sustain a host? And so what I'm curious is, are these microbial interactions that we have within a microbiome, how important are they? And is the diversity of these interactions is what maintains host fitness within this type of association. So the first step we've been working on is developing procedures where we can knock down the host microbiome. And to do this, we have anemones. We put them into these 6-well dishes. We have done an experiment now last year where we had 2 conditions, either artificial seawater or filtered artificial seawater, to first knock down the microbiome. And then we applied 2 different types of antibiotic treatments. And we watched for their recovery for 1 week following that exposure. So what we were able to do with these knockdown experiments is first significantly reduce the microbial load within these different treatments. And that's shown by these red points across our different experimental time frame. One thing to note was that upon recovery, they were still mostly germ-free, although one treatment did a little bit better than the other. We also see when we knock down this microbiome that we have a reduced in overall total protein. We also see a reduced in algal cell densities between our treatments. And finally, we see that the individuals who are treated with the antibiotics are not able to produce clonal offspring in the antibiotic treatments. And this is probably because there's just this reduced, they're having a reduced amount of effort that they have into their replication processes. And finally, we see that overall the antibiotics have this shift in phenotype. And interestingly, the different types of antibiotics have a different type of induced phenotype. So we are currently working right now on analyzing 16S rDNA sequencing libraries. And my technician is leading that effort along with the undergrads that were in the lab at the time. So our second step in this project is to begin to cultivate microbial species from this complex microbiome. Right now, we have over 75 cultivars that we've sent off for 16S rDNA screening. And we hope to expand the different types of cultivation medias that we are using to be able to cultivate a broader diversity, with really this goal of trying to capture the most prominent members and the most diverse members of that association, so that we can begin to really address this question to explore metabolic cross-feeding within the host. So I also want to tell you really briefly for the last 1 minute of my talk about this other project that we have that begins to incorporate this aspect of the environment into our understanding of these microbial interactions. And I, along with a group of researchers here at UC Merced, were recently awarded a BII grant to found this institute called INSIGHT, or the Institute for Symbiotic Interactions, Training, and Education in the Face of Climate Change. And our overarching question for this institute is, how will climate change impact beneficial partnerships between animals and microbes? So we are working with 3 different study systems to try to explore this question. Of course, we have my Aptasia system, which is this complex microbiome that we are denoting as a symbiotic association of complex. We also have a simple system, which is the squid vibrio symbiosis. The squid are super interesting symbioses because they pick up their microbial symbiont, and there's only one type of symbiont in this case, every day from their water column. And then they expel them, they pick them up at night and then they expel them during the day, and they use it for this camouflage defense. And then finally, we have a leafhopper system, which is an insect that has two different types of symbionts, but they're so interlinked with this host that the symbionts can't survive outside of the host, and the host can't survive without the symbionts. And this symbiont association is what we consider closed. So our goal with this institute is to collect a range of different types of data that really explore the fitness, physiological, molecular, and genomic response of these different partnerships to global climate change stress as induced by temperature and ocean acidification conditions. And we plan to do this with both short-term acclimation experiments and long-term adaptation experiments. And then take the findings that we see and try to validate some of our measurements with field-based populations that are under this natural gradient in terms of associations. So in addition to the biologists that we have on our team, we're actually working with a number of applied mathematicians. And one of the really cool things about being at UC Merced is that we have an applied math department. We do not have a pure mathematical department. And what the applied math people are really interested in is developing new approaches for investigating and integrating these different types of diverse data. So we have our temporal scales and our biological scales, and the goal is to generate models that can accurately capture the response of these different symbioses to climate change analysis. And we are working to help or to integrate these findings with conservation efforts to help prioritize the types of data that we collect, to generate population level species models, also to develop these resilience maps, which can help us predict how symbioses will face in a changing climate. And of course, to train and educate the next generation of symbiotic researchers. And what's super exciting is that this project is super interdisciplinary. We have everything from chemistry to applied research, and we plan on leveraging these different team science and mechanisms to enable co-mentoring and cross-training and peer-to-peer collaborations of our students. So I hope that you have appreciated a little glimpse about how we're going about approaching this really complex problem of how symbiotic associations are interacting with one another and being mediated by the environment they live in. And I want to thank you once again for your attention and for the invitation to come and speak. I'm thanking my lab members and the folks who worked with me on my project at the Max Planck Institute. And I'm happy at this time to have any questions.
Does anyone in the live room have any questions?
Hi there, thanks for the really interesting talk. I have a couple questions for you. The first one is, with respect to the first part of your talk, do the terrestrial relatives of these sea grasses also release sugars into the soil in a similar way? Is it the same kind of sugars?
I think I got part of it. If terrestrial relatives also secrete sugars, yes, they do. Usually the primary photosynthate is sucrose is glucose in terrestrial species. One of the primary differences between terrestrial ecosystems and the marine ecosystems is the abundance of oxygen within the soil environment. And so that could be one reason why they're not accumulating at such high levels.
Cool. And my second question was about aptasia. What is a meat organism? What can you tell us a little bit about? So you're rearing them in the lab, you're doing a little anemone husbandry, as it were. How long is the generation cycle on these things?
So this is a good question. We are able to get them to divide asexually in the lab, and you can force them to do it once they're like, I don't know, have a centimeter or even less of a oral disc, which is basically the part of their body that attaches to the wall of the tanks. I use tanks loosely. You can actually cut them in half and you can force them to generate new ones. But in addition to that, they will actually leave little pieces of their tissues on the side of the tanks. And from those pieces of tissues, they will grow anew. And so we think if we wanted to or needed to, we could double the population within 2 weeks of having an individual.
And can you force them to go sexual?
So folks in the aptasia community have tried with the sexual reproduction, and you can get them to spawn both sperm and eggs. However, the problem is, and you can get them to form gametes. The problem is folks haven't figured out how to get them to settle within the lab. And so that's like the major bottleneck right now. And I don't plan on tackling that issue. I think there was Jonathan Pringle was at Stanford, who's a person who started the aptasia model system, and he said they had a grant at one point and just it didn't pan out. So hopefully somebody someday will have the answer to that question, because I think it could be a really useful resource.
Very good, thanks.
Yep.
Hi, thanks for that really interesting talk. My name is Karen Gilliman. I was just curious, you had alluded to the exceptions of certain microbes that could use sucrose in your communities. Can you tell us a little bit more about those?
Yeah, so we went and investigated different types of, we went and looked at the genomic investment into transcriptomic resources that different MAGs had, or different microbial genomes had within that ecosystem. And what we found was that some of these genomes had more transcriptomic investment for degrading sucrose than other types of sugars that were in the Caszyme database. So I can try to give you a picture of what this looks like in terms of this analysis. Can you see that? So what we had is that the like some MAGs, like for example, this one had more resources invested into transcriptomic of degrading sucrose than other sugars, and that include all different types of Caszymes that were annotated in the MAG. And really interestingly, these MAGs that we denoted as sucrose specialists also had the genes to degrade phenolics under anoxic conditions. And so what that tells us is that the MAGs that are able to degrade those phenolics that are in their immediate surrounding also could use the sucrose in their metabolism. Did I answer your question?
Yeah, thank you.
Does anyone on Zoom have any questions? You can unmute or put a question in the chat. Anyone else? Questions? What's that? Okay. We're also going to have tea time at 11 or 1:15 to 1:45 on Zoom using the seminar link if you want to speak with Dr. Sogin more.
Thanks.
Yep. That was a really awesome talk.